I would like to start by thanking the organizers for inviting me to come here and talk about my research. I'm very excited to see the other talks too, and also have the discussion after. So I'm gonna be talking about the research that I've been doing here at Syracuse University for the last couple of years. So this here is a picture of my first field site back when I was in college in Brazil. And this is a type of savanna from Brazil. But this site in particular was very disturbed. So it was all this grass here, most of it are invasive species. And we can see some of the plants are kind of burnt because this site usually goes through fire events very frequently. And not apparent here, but the site was also right next to a huge monoculture of sugarcane. But even this very disturbed site, in this one picture, we can already spot a couple of interactions. So here I have, for example, flowers, those purple flowers. This is interaction between the plant and the pollinators. This other plant over here was the plant that I was studying during my undergraduate research and also my master's. And this plant in particular has structures that have evolved to interact with ants, and the ants protect the plant. And not very apparent here, but we know that many of those plants, the species of plants, also form interactions in their roots with microbes that help them with nutrient acquisition. So those examples are examples of a type of interaction that we call mutualism, that is a beneficial interaction between different species. And I use this example to start my talk because we can see that even in a very disturbed habitat, we can find different types of mutualisms, because mutualisms are present in every habitat. They are extremely common. We all right now are in a mutualism with our gut microbiome, for example. And plants in particular rely on mutualisms for their reproduction, for example, in pollination. Plants can also form mutualisms with animals that help them with seed dispersal. And as I mentioned, some plants can also have special structures to interact with ants. So this plant in particular, the one in the left, the yellow circles here on the base of the leaves are what we call extrafloral nectaries. Those are glands that produce nectar, but they are not related to the reproduction of the plant. So this nectar attracts ants, and because ants tend to be very aggressive, they end up attacking herbivores on the plant, that way protecting the plant against herbivores. And plants can also serve as a substrate for other mutualisms as well. So many times when you're looking in plants, we can see those sucking herbivores, which are similar to aphids, and ants walking on top of them. And this one here is also a scale insect, which is similar to an aphid too. So those sucking herbivores, they produce a sugary droplet that attract ants, and the ants also end up protecting those insects from their natural enemies. So also it's a protective mutualism. And we can think of mutualisms in general as this interaction between species where those species are exchanging resources or services that they need, but this usually comes at a cost for those species. So in pollination, for example, we have the plant producing nectar, and there's a cost to produce that nectar that will attract the pollinators. that will benefit from this nectar because nectar is a source of food for them. And in return, the pollinator will transfer pollen from one plant to another. And that can also have a cost to those pollinators because they can carry sometimes pretty heavy loads of pollen from one plant to another, which takes some energy for those pollinators to do. So usually we have this conflict of interest between those interacting species. They are trying to increase as much as they can the benefit that they get from this interaction while reducing the costs that they will have. So in the extreme of this conflict of interest, we have what we call cheaters or exploiters, which are species or individuals that take the resources service that the partner is offering, but without giving anything in return. So they completely, they don't have this whole cost side of the interaction. And those cheaters or exploiters, and I'm gonna be calling them cheater or exploiter interchangeably, they are very common in agrimutualism. So for example, many times in pollination systems, we have what we call nectar robbers, which are animals that make holes on the side of the flower so that they can directly access the nectar without even touching the pollen. So they are not transporting pollen from one plant to another. And those cheaters can have negative effects to the plant because many times they end up depleting the plant of nectar. So the flower is not gonna be attractive to other pollinators. And they can also have fitness advantage over the real pollinators because they are not carrying that load of pollen from one plant to another. So in my research here, I want to understand how can mutualisms persist with this constant threat of exploitation. So many times in mutualisms, what we have are very diverse communities of many different species interacting with each other. So for many pollination systems, for example, we have different species of pollinators that visit different species of plants. And this species richness or number of species that we have in a community can be protecting those communities from exploitation. For example, if we have different species of bees visiting the same flower, if one of those species is removed from the interaction because of competition with a cheater, for example, the other species that remain can still provide the service to the plant of transferring pollen from one plant to another. So this mutualism can persist even with the exploitation. So usually in ecological research, we tend to do a lot of field work and also using natural systems, which are great ways to understand how natural systems work. But many times we have some challenges with those field work or natural systems because we end up studying long-lived organisms like many trees here, like this one, for example, that we can only get like a couple of generations in a period of time that we are doing that research. And we also are limited on how much we can control or alter different factors to address our hypothesis. So when I joined the lab of Carrie Seagraves here at Syracuse University, I was interested in using this yeast synthetic mutualism that she was developing. So what we have here are yeast, so the same yeast that we use to make bread or beer, but we genetically modified them to produce more or less of resources that they need. So I'm gonna be particularly talking about lysine and adenine, and the only thing that you need to know about them is that they are basically a source that they kind of like food for the yeast. They need those resources to grow and function as a cell, and also to divide and reproduce. So we have our adenine overproducer, which produces more adenine than it needs. So it releases the excess to the medium, and it cannot make lysine. And we have our lysine overproducer that produces more lysine than it needs. That's also released into the medium, but cannot make adenine. So they are exchanging lysine and adenine that they need. And we also have cheaters. So we have a lysine exploiter that produces adenine only for itself. So it's not contributing to the mutualism, and it cannot produce lysine. So it is competing with lysine with the adenine mutualist. And when this exploiter wins the competition and removes the mutualist from the interaction, this mutualism, we call that it breaks down, because all of the other species or yeasts that will be involved in this interaction will not gonna be able to survive without the adenine that the mutualist produces. So when I talk about a community survival, I mean that the whole community died, both of the mutualist types and also the exploiter. So to look at how species richness could make those communities more resistant to exploitation, we created communities with different numbers of species of those yeast mutualists. So we started with our simplest community, which is the one here on the left, where we only had one species of the adenine mutualist and one species of the lysine mutualist. And we increased the numbers of pairs up to our most complex community with 4 species of the adenine mutualists and 4 species of the lysine mutualists. So we had those communities with only mutualists, and also communities where we included the lysine cheater. So we let those species grow together for 4 weeks, and then we assess how many of those communities had survived. So here on the x-axis, I have the initial number of mutualist species that we set up those communities. So as I mentioned, it goes from 2 species or 1 pair up to 8 species or 4 pairs. And in the y-axis, I have the proportion of the communities that survived until the end of the experiment. So in blue, we found that all of the communities that only had a mutualist survived until the end of the experiment, while the communities where we included the cheater, we can see that they suffer up to 60% breakdown. So this means that all of the species were removed from those communities. But we can see that the survival of those communities increases as we increase the number of species that we include, mutualist species that we include in those communities. So this shows that species richness can increase the persistence of the survival of mutualisms under exploitation. So considering that most of the mutualisms that we have are those diverse communities of mutualists, they can be inherently buffered from the negative effects of exploitation. But what about the pairwise interactions, those simple one species interacting with another species, mutualisms? How can they persist also with the exploitation? So although they are not as common as those diverse communities, those pairwise interactions do occur in nature. And a great example of that is the one between the mutualist between yucca moth and the yucca plant, which is a mutualism that Carrie has been working here at Syracuse University for many years already. And it's also a model that we use to understand how mutualisms evolve and how they can persist with cheating. So just to summarize how this interaction happens, because it's also very interesting. The yucca plant, usually only one species of the plant interacts with one species of the yucca moth. So they are very specialized. And the yucca moth, they have special structures to collect the pollen grain. And we can see that in this moth here carrying the pollen grains. And they insert the pollen in the flower, so fertilizing the flower. And after they're done with that, they lay their eggs inside the flower. So the offspring of those moths will feed on the seeds on the developing fruits. And we also have a cheater. I don't know if you can see over here. This is a moth that instead of pollinating or fertilizing the flower and then laying their eggs, they actually skip that whole part and lay their eggs already in the fruits. So after the flower was fertilized by the pollinator. So in this example, we have those structures of the plant and of the moth that had evolved in response to one another. And this reciprocal evolutionary change between those interacting species we call coevolution. So I wanted to see if coevolution could be making those pairwise mutualisms more resistant to exploitation. So to do that, I set up cultures where I only included one species of the adenine mutualist and one species of the lysine mutualist, as well as cultures where I had those mutualists and the cheater. And I let them evolve together for over 300 generations. So after those 300 generations, I look at how many of those cultures had survived. And I found that, similar as to the richness experiment that I already talked about, all of the cultures with only mutualists survived to the end of the experiment, while only 35% of the cultures with the cheater that started with the cheater survived. And of those cases of exploited mutualisms, the ones that survived, the large majority of them are cases where at the end of the experiment, we only had mutualists. So the mutualists kicked out the cheater from the interaction. So to see if those co-evolving mutualists could be resistant to exploitation, we got the yeasts that had evolved together for 300 generations, so they already are co-evolved, put them together with different types of cheaters. So we had the same cheater that had evolved with those mutualists. We had cultures where we put the ancestral of that cheater. So it's the same genotype as the cheater that evolved with them, but without going through those evolutionary changes. And we also had what we call a novel cheater. So this cheater has a different function in that interaction. Instead of exploiting lysine, this novel cheater is exploiting adenine. So we let them grow together for a couple of weeks, and then we look at how many of those cultures survived and which species we had that was still alive. So here, this is the same figure that I already showed before, the results after they had evolved for 300 generations, just to remind you that for that experiment, the cultures that started with the cheater, only 35% of them survived. But now that we included, we started the experiment with already co-evolved mutualists, 96% of the cultures survived. So this is already evidence that those co-evolving mutualists might be more resistant to exploitation. And we also look at how many are the percentage of cheaters that survived with the mutualists. So we found that the evolved cheater survived with the co-evolved mutualists in all of the cases. So this is showing that potentially those cheaters are also co-evolving with the mutualists, while only 12.5% of both the ancestral and the novel cheaters survive with the mutualists. So this means that in almost 90% of the cases, the co-evolved mutualists are removing the cheater, the ancestral and the novel cheater from the interaction. So this means that co-evolved mutualists are resistant to exploiters that don't share evolutionary history with them. So considering that exploitation or cheating is extremely common and present in every mutualism, but mutualisms still can persist with this cheating, we have shown that those intrinsic factors of mutualisms, that being species richness or coevolution between partners, can be what is making those mutualisms resistant to exploitation. So I'd like to thank the Species Interactions Lab here at Syracuse University, and also invite you to check our Species Interactions Lab Instagram if you want to see more pictures of bugs, and also our funding agency.
Thank you, Moira. That was really nice. So next up, we have Mate.
Hi. Can you hear me, guys?
Yeah.
Great. Okay, so let me just start the screen share. Okay, and oh. Okay, and let me just hit play. You guys can see my slideshow, right? Or, oh no, that's presenter notes. Oh no, 1 second, sorry.
We just saw the slide.
Okay, 1 moment. What do you see right now?
We see your name.
Oh, my name, perfect. Okay, yeah, that's how it should be.
Yeah.
Okay, great. Yeah, hi, so my name is Matej Vakula. I am a PhD student at the Rensselaer Polytechnic Institute, and I work with the KP High Lab, or BAT Lab at Center for Biology and Interdisciplinary Studies at RPI. So first, I would like to show a couple of older works too, just to set sort of the mood and very quickly, and then I will start talking about the core of the presentation. So let's go. So this work was done in 2015, and it's called Well Played Utopias. Around that time, I worked with the Marion Sloan Kettering Cancer Research Center, with Daniel Heller's lab, and I worked with tissue culture and testing their drugs, the cancer drugs they are discovering there. The interesting point about it is that they are using artificial intelligence to sort of design the drugs, and then humans test it and see if it works or not. And so these are those drugs, and I sort of figured out a system where I used the major feature of the drug is that it is light activated, and after it is light activated, it kills the tumor cells. So I was sort of playing with that, and I utilized that process in sort of as an analog photographer would use photo paper and a film. So I used film, but instead of photo paper, there was the well plate with the cancer tissue. And I projected this weird kind of alphabet on the well plates. Here you can see it, sort of a mock-up for installation on the wall. And so it is instead of 24 letters, it's 22 letters. And this alphabet comes from Thomas More's book, Utopia, the book from 1600s. So this was the written language of the island of Utopia. Thomas More made up also the language and also a new alphabet for them. And I used this alphabet as sort of messaging technique where the patients could send messages in these letters to the lab, kind of involving their thoughts on their research. Because usually just the lab has, pushes new discoveries and like sort of creates the future for the society, but the society somehow has to deal with that, cannot really often reply back. And this is kind of nice that the patients had the chance to reply. Second thing, what we did together with the same laboratory is that, again, as I mentioned, they work with this drug discovery using artificial intelligence, where this software proposes these drugs, where I just have shown the effects of these drugs on the tissues, but here are the drugs themselves. And basically, these are supercomputer simulations using artificial intelligence to mix these regular cancer drugs with nanotubes and nanotechnologies, and that is supposed to create a targeted photothermal treatment for cancer. There's one very interesting thing happening where when you use artificial intelligence or in particular machine learning to help you to discover stuff, it often you end up with a working result, but you don't really know as a scientist how the machine learning software exactly came up to that discovery. And so it is closely related to the complicated nature, the way the software works. Sometimes for a human mind, it's almost like if you took a scan of a brain, a slice, you cannot really find the thought in it. There will be just some sort of points that connect together. So it's hard to really tell, because there are millions of interactions every second. So we together came up with a kind of awkward solution, but it was interesting that it was actually suggested by the scientists that they would like me to design a tangible sort of construction set, almost like a Lego, made out of those molecules, and so they can play with them with their hands. And that will help them more in the physical world to understand the interactions. There are some magnets inside these models, and so I simulated very basic Newtonian physics that were connected with those particles. And I mean, I'm not sure if it helped, but they really liked it, and it was more like a supportive sort of thing, springboard for their thinking, I would say. They said it helps, but I think it helped more in the level of sort of your body has to understand things, not only your mind. You have to understand things in the 3D world. And there are some papers on interaction by Natasha Myers, for example, exactly with this molecular modeling technique, that your body has to somehow connect. It can not only be the computer, you have to understand the data tangibly sometimes. Yeah, that's a couple of those with Dan Heller. Okay, so after I kind of or showed the direction where are we going, which is the artificial intelligence and machine learning, how it is involved in science, and sometimes how does it help scientific discovery. You can see that I'm sort of looking for a little bit different ways, because I'm artist, of how to utilize these scientific technologies. And so I would just like to say or put out a disclaimer in a way that, I am not with my works, although these were a little bit more particular, but I'm not making claims that I'm saying the truth or truth claims. Rather, I like to use speculation or fiction where I examine other truth claims around some ideas related to science. So I like to look at culture that is connected with certain scientific ideas, and that is supported by many papers in science and technology studies. For example, we can start with Thomas Kuhn, The Structure of Scientific Revolutions, or Karen Norsetina, her whole work, or Ludwig Fleck, Genesis of Development of Scientific Fact, for example. Yeah, so I like to create fiction based on science, which is science fiction. These two guys are really interesting for one very odd reason. So Stefan Leduc coined the term synthetic biology, and Jacques Loeb sort of created the first engineering paradigm in science, where he pretty much the first time, he's the one who stated that understanding equals the ability to build. So basically, he believed that if you discover some idea on observation, but unless you can rebuild that thing from that observation, it is not a scientific fact. Which is interesting because to me, philosophically thinking about life, for example, we cannot really yet build completely new life or completely new life form. Even though we have fairly advanced technologies, we would need to be probably way, way more advanced as this right now. So this is work of Jonas Zur and Oron Katz, and they actually reacted to Stefan Leduc because he actually believed, as I mentioned, he was the father of the term synthetic biology in the break of the century in 1900. Or around the year 1900, and at that time he believed that he created artificial life, and he wrote a book about it. And basically he started working with protocells, where he believed that he based those protocells more on how it looks like than actually what it does. And so he used a mixture of ink and, I believe, soap, where he was dripping drops of that into the water, and it started, or into certain kind of suspension, where it started creating these cell-like structures, and sometimes they created some more organic shapes. And that's why almost like corals sometimes, and he believed that this is actually life. So of course he was proven wrong later, but the term synthetic biology stuck with us, and it evolved, and right now, of course, scientists don't do these claims anymore, this type, and but you know, we are still trying to invent life. We are artificial life, basically. And so if we think about what is life, or how are we trying to create life, we are trying to recreate it nowadays using computation, or we are trying to gain knowledge about life just by observation. We can sort of think about both of these types as black boxes. Biological life itself is a black box that we are step by step unraveling, discovering, clarifying, let's say. But it is still quite a bit of black box. We have not been able to create like completely independent artificial life from scratch. Also, and then, but we are pretty good with computation and computational simulations of life. But when we are using machine learning or artificial intelligence to create life, again, it becomes a sort of a black box where we don't know exactly how, where we don't know exactly how the machine came up with particular results, and then we have to sort of backtrace those ideas through those algorithms. So in a way, right now, we are in a situation where we have two black boxes next to each other, computation and biological life, and they are sort of starting to influence each other. We are starting to think about the biological life as a machine, and we are trying to think about computation because it's more and more black boxed. We understand it less and less as something alive. So these kind of intertwine. Now, what are the ethics of these discoveries? I mean, if we don't know exactly what it is, if both of these things are still black boxes, and the life may be less now, the bioethics is still kind of like we're trying to talk about ethics of that black box. And then also we are trying to talk about ethics of that the other computational black box. So yeah, so in a way, what are when we're trying to create new life, there are two approaches. There is a top-down approach that is pretty much let's scan, let's make the most precise scan of, let's say, brain, and then we will be able to recreate it in computer. And then we should be also able to, again, recreate it or 3D print it in real world. And then there is another approach, bottom-up approach, which is about this recreating it in the real world. So the top-down is scanning and putting it into computer. The bottom-up is recreating it from scratch, basically. So from the bottom-up approach, for example, there is work of David Benjamin and Fernand Frederici in where these guys scanned xyleme structure of a plant cell, and then they extracted the core algorithm, or I could say like quote unquote growth algorithm, and using machine learning, and then they can apply this growth algorithm to different types of cells or different architecture, even architectural forms. So there is always with machine learning, there is some sort of extraction of some sort of a quote unquote substance. I'm not saying a gene or genetics. This is, even though it is called confusingly genetic algorithm, this is more about creating sort of an essence of growth, not only from genes. Or maybe sometimes genes don't have to be even involved. It's more from the scans, from how it looks like. And then once the machine sort of figures out the quote unquote essence of growth, it can produce things. Like, for example, in the works of Sofia Crespo, she's playing with this idea where she extracted growth algorithm from some beetles, and then she is letting the generative algorithms of machine learning to go wild and create these weird beetles that never existed before. Of course, this is only in the computer. This is not live recreated. But this is how generative modeling works. Now, from the top-down approach, there is this interesting project from Europe called Blue Brain, where they are trying to recreate, trying to create a scan of the brain, up to date, the most precise replica, electronic replica of the brain. They found out that, or their theory is based that in the idea of neocortical columns, which is for them sort of the basic building block of the brain. It is a small column, couple of micrometers wide, that contains a couple of hundred neural cells, neurons with thousands of synapses. They're trying to precisely scan these synapses and then recreate the way how they work. They completely scanned a mouse brain, and they are working on a scan of the human brain. Of course, for me, there is now this question, like, you know, with scanning, there is also the problem that what is scanned and what is left out. So, I mean, is it really a good question? Like, even though this will be the most precise model of human brain, I would say that it is very hard to tell that, you know, if they will be able to encompass all the options that exist in the real world, because of course, every model is a simplification. But it is interesting that around this idea, there's growing sort of this movement. It is at the core of the transhumanism, or in other words, H+ movement, with, for example, Ray Kurzweil in the forefront of that, which pushes this computationalist idea of the brain, basically that we will be able to recreate our minds in the computer. Now, I'm just thinking about that and, you know, comparing it to Stefan Leduc and his idea to how he tried to recreate a life 100 years ago. And it is, you know, it is again rather cultural phenomenon around it, because we are not there yet. So we can only imagine the future of this technology. So again, what could be the ethics of this? Now we're getting into sort of expansion of the human abilities, and at what cost, and also what ideas about the mind are involved in this. Is mind really a machine? Is intelligence everything that mind does, or is it not? For the transhumanists, the rupture, there is the rupture between animal and human. The human is in the near future should distance him or herself from the animal, and then exponentially grow with help of technology. But I think that is kind of opposite to actually what science is telling us, because science is telling us that we are actually we are animals. There's not that big difference between, biologically speaking, between even within mammals, for example. So should we actually think about life and simulation of life that is geared only towards the human, or as human as the pinnacle, as the top of the world? Or should we think about more non-human ethics? Because if we put the human in forefront of everything, so then we can see that from our history that we sort of don't consider animals or plants or the environment as an co-equal thing to coexist with us, rather something to use to our advantage. So there is really interesting thing that or direction in ethics that the recent direction in ethics, which is called non-human ethics, and that's what I'm kind of trying to research, looking at these technologies. Also, sometimes we look at artificial intelligence as anthropomorphic, but it is not. It is more infrastructural. It is something that is not a friendly robot. It is something that is more like an infrastructure, so, or a data visualization infrastructure. So people often forget about it, and especially the transhumanism or the H+ movement is trying to present robots only as those human figures, in a way, kind of a cliche robotics, which is not, again, preventing us from experiencing the other side of that. So these are some examples of the neocortical columns, and on the right side you can see neocortical, those beehive large parts are superstructures, which are called, those are actually called neocortical columns, and those small lights are called neocortical mini columns, and that is the basic building block of quote unquote mind. So what are the ethics associated with these technologies? Can we use human ethics, or are there better alternatives to research these sometimes inaccessible black boxes? We are looking at non-human ethics as the possible closest thing that we can use to research these black box ethics. And I would like to create sort of in the lab so-called non-human ethics implant, which supposed to provide non-human ethics to implant for the human brain. It's supposed to help us to think about the environment, and it's supposed to provide us with sort of more compassion to the animals. Of course, this is a sarcastic kind of idea, but I am planning on actually growing human brain neural stem cells together with the animal stem cells and create sort of like machine learning the same way is working, that the learning is based on rewards. It is called reinforcement learning. So I will create this organ where human brain stem cells or human cells are connected with animals, but the human are turned into dopaminergic neurons that will provide the rewards in the reward system that brain is using for the animal. So this reconstruction of learning about ethics will be connected to the AI algorithm that will also intervene in this learning, and it should help us to think about non-human ethics. So here is the system, how it is going to work. So the model will be generated using these neural databases with generative adversarial network, which is machine learning network that's supposed to model, create a model based on what it learned from how it grows, how brain grows. And also I would like to connect it with the machine ethics module, which supposed to sort of inject ethics into generating that that implant. And part of that machine ethics will be will come from database with bioartworks that are dealing with non-human ethics, and also live evaluations of this model in a feedback loop from the bioart community. After that, after the model is created, it will be 3D bioprinted and incubated as long as possible. I'm going to use Rensel's human neural progenitor cell line and cortical neural stem cells of a mouse. Same as in that model. Thank you.
All right, thank you, Matej. So we're gonna take a short, 5-minute break. Let's have everybody come back at 7:40, and then we're gonna have our last talk from Jennifer and a discussion.
Cool. I'll see you all in 5 minutes. Thank you, Matej. And
thank you.
Yeah,
should I stop sharing, or you guys are doing it?
You stop.
You have to stop.
Mm-hmm.
You have the power. Cool.
Okay, so I'm going to set my timer in 2 10-minute increments. I am going to talk about micro and macro landscapes. And the outline of my talk is first science, and then art, and then science and art, if I can pull it all off. In the beginning, I'll talk about the Carbon Farming Report, which we just released. It's a project that I've been doing with Peter Woodbury at Cornell for the last couple of years. Talking about what is carbon farming, what we evaluated, and our top ranked opportunities for mitigation for New York State. And then I'm going to talk about my mud paintings, which are micro landscapes. And then if we have time, I will talk about my new science-based art, or art-based science, depending on how you look at it. So the first thing I want to talk about is this report we just released in May. We fondly call it the Carbon Farming Report, and it was supported by the state of New York and the Department of Ag and Markets. And it's following some New York State mandates that are listed here, and I'm gonna skip. But one of the most important things that everybody in New York State should know about is the New York State Climate Leadership and Community Protection Act, or the CLCPA, which passed in 2019. And it has magnificent and very exciting and very ambitious goals of a 40% reduction in greenhouse gases in New York State by 2030, 85% reduction in greenhouse gases by 2050, and it applies to all sectors, including agriculture. And so I have been focusing on agriculture, and as you can see, agriculture contributes about 4% of the state, but we still have to meet those same goals that everybody else has to meet. So what is carbon farming? It was defined by the Carbon Farming Act, which was passed by the House, A3281, and it was defined as, I have to reduce, the implementation of land management strategies for the purposes of reducing, sequestering, and mitigating greenhouse gas emissions on land used in support of a farm operation and quantifying those greenhouse gas benefits. So what we did in this report was we looked at sources of emissions and mitigation potentials from different farm activities. And the first thing you should know is that while most of society has to think about CO2, farms have to think about 3 greenhouse gases. I'll talk about them in a minute. There are some general mitigation strategies, which I probably will skip, but we focused on 13 different mitigation potential opportunities, and we qualified that mitigation with co-benefits, like does it mitigate greenhouse gases and improve water quality? Does it actually save a farm money? Those kinds of things. The scale of the opportunity, how big is it across the state? Is it a large opportunity or is it a small quantitative opportunity? How much does it cost to implement on a farm? Is it $10 a metric ton or is it $150 per metric ton? Is it realistic? Are there like 9,000 stakeholders with very different opinions on things, or are there 500 farms and a huge opportunity with technology that is currently available? And then how permanent is this mitigation? Is it like me eating a tomato and respiring the CO2, meaning it's not permanent? Or is it something like growing carbon in a tree and that grows for 100 years and then cutting down that tree and having it be a building material that lives for the next 200 years in a house structure? One of the things I just want to impart to everybody is that while CO2 is the most important greenhouse gas because it comes from fossil fuel, which is the predominant issue with climate change, in terms of agriculture, we have to think about methane and nitrous oxide. Those are 2 very potent greenhouse gases. And so this idea of carbon farming is something of a misnomer because it involves both carbon dioxide, carbon-based methane, and nitrous oxide. And the reason why they're important is that they have different potencies. So methane is 84 times as potent as of a greenhouse gas as carbon dioxide, and nitrous oxide is 264 times as potent. And I say this because what that means for agriculture is that 75% of the greenhouse gases in agriculture come from methane and nitrous oxide. And so particularly, these are the things that agriculture needs to focus on more than the carbon dioxide. These are the 5 main categories by which you can mitigate greenhouse gases. You can sequester carbon in trees and long-lived wood products. You can destroy the methane. So the methane that is in the manure storages can have a cover and then a flare, and that destroys the methane. You can increase the efficiency, which means you use less energy on the farm. You use less animal feed to produce your milk, so the milk is more efficiently produced. You reduce how much nitrogen you apply to the land, and therefore you reduce the nitrous oxide emissions. Another thing you can do is to displace fossil fuels, either by growing solar energy on your acres or by growing biomass. There's lots of different ways that you can displace fossil fuels. And then there's always the most incredible and effective measure of mitigation, which is conservation of both energy and natural resources and leaving forests as forests, for example. So the goal of the project was to determine effective practices for agriculture to mitigate. And these are the 13 practices we evaluated. I can go on and on about this, but because of time, I'm going to tell you about the top 5. Manure cover and flare, there's about 500 large manure storages in New York State. And if we just simply put a cover on those manure storages and collected all the methane produced by methane producing bacteria and flared it, we would have a wonderful opportunity to mitigate emissions. Nitrogen use efficiency in the 70s, and even recently, people would put nitrogen insurance, which is extra nitrogen on their field to increase their growth of whatever crop they're using. But excess nitrogen often comes off as nitrous oxide, which is the very potent greenhouse gas. And so if we can just get more efficient in using our nitrogen, that is a fantastic opportunity. If we improve the feed efficiency from our, especially our ruminant animals, we produce less methane from those living animals. If we improve the management of our woodlands, that is, we keep them healthy and we promote their growth as opposed to sort of letting them go wild. That's a whole other discussion, especially with this group. And then we could think about planting trees on idle lands. We could think about putting solar on idle lands. We could think about all kinds of things, activating the 1.7 million acres in New York State that are currently somewhere in transition between agricultural lands and forests. And briefly to summarize, on the left-hand side, you'll see the emissions total from our estimate of emissions total from all of New York State agriculture. It's 9.9 million metric tons of CO2 equivalents. And those top 5 mitigation strategies I just mentioned, which we estimate to be under $50 per metric ton of CO2 equivalents, those top 5 opportunities would reduce 7.1 million metric tons. So right there you have basically 70% mitigation potential. So just to recap, remember the CLCPA is a 40% reduction in greenhouse gases by 2030 and 85% reduction of greenhouse gases by 2050. And so, you know, these 5 opportunities can basically get us mostly there to that 2050 goal. Is everything a win-win? No, but there are always good choices. And our next project is this April, we began a 3-year project with DEC to create a more comprehensive New York State agricultural greenhouse gas inventory, mitigation potential, and 3 future scenarios. And we'll get back to you. This has been supported by the Department of Ag and Markets, Department of Environmental Conservation, and NYSERDA. And we have a blog, and of course, my colleague, Peter Woodbury. And these are the 2 key references. Look at that, 9 minutes, 10 minutes, right there. Okay, science-based art. So I started making art based on science in probably realistically around 2002, and I'm gonna start showing a project that I fell in love with from 2004 to now, and I love them. So Winogrodsky Rothko was my first mud painting I made in 2004, and it was inspired by a summer fellowship where I was a TA at a marine biology program for high school students at St. Paul's School in New Hampshire. And we made a Winogrodsky column from some marine mud. And the Winogrodsky column was inspired by Sergei Winogrodsky, who lived in the 1800s in Russia. He traveled around also. And he made these columns like you see here on at least my right of the screen. And all of that pigment you see in there is from living microbes photosynthesizing pigment. And this was just after we invented the microscope. And Sergei Winogradsky was very interested in soil, and he thought there was something incredible going on, but he had no way of knowing. So he put this column of soil in his window, and he waited while he was working at his desk. And one day, 3 weeks later or something, he sees a red splooch. He puts his pipette down into the red splooch. He puts it in his new microscope, very exciting technology. And he sees soil particles, which are stable, and then he sees these moving particles, which were microbes. So this is early soil microbiology. And what he and many others after have determined is that there's all these relationships of waste and resource utilization flipping along these different layers, these horizontal layers, which are ecological niches. And these horizontal layers happen because at the top you have lots of oxygen, and at the bottom you have very little. And at the bottom you have lots of sulfur, and at the top you have very little. And so what that means is that as you move down the column, right, you have a highly aerobic, low sulfur concentration, some oxygen, some sulfur concentration. And at the very bottom, you have lots of sulfur and almost no oxygen or no oxygen. And what that does is it creates all these horizontal niches that select for all these different bacteria that are just amazing in their capacity to do stuff with stuff. So I guess I was 23 when I did that program at St. Paul's with high school students. And while we were doing that, I was reminded of my first Rothko painting in 4th grade when I took a field trip to the Hood Museum. And I saw this painting, and look at all those horizontal bands. So in 2003, I got a grant, and in 2004, I made a frame in the size and dimensions of this Mark Rothko painting. And this was my first series of mud. It's 3 feet by 5 feet. I actually had to build it twice because the first one broke because the water pressure from the glass was so great. But here you can see this is a 3-month process. And you can also see I was learning photography. And in fact, these are photographs by John MacArthur and, oh no, Kathy, I forgot your last name, but I'll remember it, I apologize. And these were some of the micro ecosystems. So in the bottom left-hand corner, you'll see the whole large frame, and then you'll see a little square indicating where these different ecosystems were happening. And what's so fantastic, obviously, is you see this pointillism that grows throughout. And one of the big lessons I learned from this, there was this gorgeous spot of like these lavenders and pinks and silvers and greens, and I was so happy. I was so happy. And then there were these snails, and they came in and they ate it all away. And then these worms, and it's just one big gigantic recycling center. And so even though I was loving like the beauty of the aesthetics, I was forced to recognize, like the rest of us dealing with climate change, that there is just so much change everywhere all the time. I was working and I didn't have much time to do my art, so the next mud paintings didn't happen until 2012, and I did a series called Portraits of New York City. And this time I chose 5 of the most polluted places in New York City. Here I am collecting mud in the Gowanus Canal. I think it's considered the nation's most polluted waterway. And here's a steel and glass frame I welded, and I'm putting the mud in the frames. And here is the Gowanus Canal about 3 months. For those that don't know, a Superfund site is considered so toxic that the national government has to come in and force the local governments to mitigate. And so it's a sign of serious contamination. But what I learned, so this down here in the bottom left-hand corner is roughly the color of the pigment of the soil that went in, and all the other pigment you're seeing is microbial action. And so this is an image in the studio. This was a Lower Manhattan Cultural Council residency on Governor's Island. This is the Hudson River, Superfund site of PCB contamination. This is Newtown Creek, a Superfund site of oil contamination, and then the Gowanus Canal, which is, you know, the kitchen sink of contamination. What I learned from this project is like microbes live everywhere, and their idea of toxicity is very different than our idea of toxicity. And I started having real hope for the world, even if it didn't include humans. This was the Gowanus Canal that I converted into a dress for the runway. I called it Drop Dead Toxic. And I would do more dresses if I had more time, but I don't. I collected mud from the East River, which had, and this is an oyster river collection project, or an oyster remediation project. So there were some oysters, which turned out to be quite stinky. I'm not recommending you put oysters in your Winogradski column. But here they are in the first 2 weeks for the East River, and then here's the final. And the reason why they're growing oysters there is because there are so many sewage overflows from the sewage treatment system in New York City when it rains a lot. And so this is a painting of New York City's excessive flamboyance with their sewage. I also collected mud from Dead Horse Bay, which is this exposed landfill down in, it's actually Queens, but it always feels like Brooklyn. And I just would note that as of last week, they determined that there's radioactivity here, so you're no longer allowed to go to this exposed landfill. But this is the mud painting that I made from that then. And here I am collecting mud from the Hudson River, where the influx of the ocean meets the downflow of the Hudson. And so there's this salt flat, or a soil flat that happens at that intersection, and you'll see my path. And the reason for this image in here is to let you know that you should always go with someone when you collect mud, because I got seriously stuck in the mud right there. Here's the results from the Hudson River, from the Hudson River site. And I went out with a river keeper to collect the mud from Newtown Creek. The river keeper is this fantastic citizen watchdog. And here's the painting that resulted from that. This is 3 months of that. And the mud is very black, and I will say it also smelled very much of petroleum. I've also done them in some of the most pristine places. This is an installation at Ciosla Model Forest, which is upstate, and it's part of keeping New York City's watershed clean. I've been commissioned for New York City Parks. This is a public bench, which I could talk more about, but I'm gonna just move on. These are 3 images from a project I did in Chestertown, Maryland. I was gonna show a video, but I think I'm gonna forego the video to talk about how my work is changing to, instead of science-based art, I am trying to make my next step be art-based science. And how am I doing that? Well, I am trying to do DNA analysis of the real authors of these paintings. And the first project happened with a collaborator named Dan Biting, who is at University of Pennsylvania. And we installed this in his basement. You can start seeing that I'm less interested in the aesthetics, the formal painterly aesthetics. And what I'm doing here is I'm grading it off, and we took these swabs from these different locations. You can sort of see the color. And then we did 16S RNA analysis. Of 40 samples, 29 yielded sufficient DNA. And then this is who was living in those 40 samples. There were over 800 species in this one painting of only 29 samples that were analyzed. Many were unnamed species. And what was most exciting was every sample had many species. So a color is actually a composite of some sort, and that has me really excited. And I also feel like Seurat, if only I could have a conversation with Seurat, it would be really fun to talk about what goes on inside of a pigmented painting. Last fall, I started a project that's totally painfully on hold because of COVID. It's called Munsell Richter. It's in process, and the goal of this is 10 mud paintings in 10 cities. The piece is currently in Bangalore, India, and it's based on Albert Munsell, who came up with a color system. This was his color system, and it was used as a soil identification tool in the 1930s. This is the soil identification tool. And so he was using color in order to classify soils. And in reference to Gerhard Richter, and in particular this piece, 10 large color charts. And so basically what I'm doing in each city is to do one color chart. And so this is sort of the mock-up of it. And the first time it was done was in Bengaluru in India. It's called the City of Many Lakes. And I traveled all around that city to collect these 10 samples in these buckets. Here they are. Here are the 10 samples. You can see without even trying how different the soils are. And the goal is to go back and do sequence analysis, but I haven't been able to go back to either photo document, let alone collect the samples. So to sort of wrap it all up, my meta question is, can microbes be a model system for us to think about equitable resource use? Because in fact, they are communities of coexistence and they're quite diverse. And I'm trying to think about how this could inform our collective policies on climate change. Like, how can we think about equitable resource use at our scale? And just excited to say that next week I am going to start a residency at Coalesce, which is super exciting for me, in part because it's the first time that I am actually formally, in sort of an art science way, formally being an art scientist or scientist artist, or I don't know. It's very exciting. So questions? 23 minutes, 3 minutes over, apologies.
Awesome, awesome. Those are a great round of presentations. Thank you all very much. That was fantastic. There's like the virtual applause. It's not quite as gratifying as being in a room. It's like kind of when Kamala Harris and Joe Biden turned to the crowd and waved to nobody, you know, in the virtual convention that happened last night. But we are somebody, we are a group here. And I really enjoyed I always enjoy the chat stuff. So my job as moderator in these events, usually there's just two people, or the ones I've done, I only had two people, and it's to find a common thread. And I was told by Boreana that I wouldn't be able to do it this time, that they're too discrete. But I think totally there's some very strong common threads. And so I'm going to start it there, but then anyone just jump in at any time. You know how to virtually raise your hand. You can do that, or you can just unmute yourself and jump in. And I'll get to some of the comments in the questions, really good questions in the thread. But just to kick it off, I found there to be two really strong themes throughout both of the presentations. And one is the theme of ethics. I think each presentation touched on ethics in a really strong way, obviously, and most explicitly in Mate's, but actually it's a kind of underlying principle in all of them, I think, to one extent or another. So I had specific questions for each of you on the topic of ethics. The other thing that I found in common was kind of this question of epistemologies or different forms of knowledge and how those kind of integrate in each of your practices. And I'm always interested in this negotiation between the poetic and scientific knowledges and these different ways of knowing. And someone in the thread mentioned indigenous knowledge as well. And I always like to bring that into, because I think we can become too transfixed with these kind of Western forms. So let's go to ethics. Myra, I wanted to start with you. One of the things I noted throughout your talk is the language that is used has a very strong kind of a moral register to it. Exploiters and cheaters and mutualisms, and there's sort of there's a clear kind of value system embedded in that narrative. And I got the drift that some of your research could be purposed to to reinforce the idea of kind of diverse ecosystems and supporting diverse ecosystems, kind of have a management, kind of have a lesson for landscape management. I just wanted to ask you though directly about how you think about ethics in your work and how you think about it supporting ethical systems, and also about that language that is used.
Okay, so the language, we're gonna start with that. There's a lot of debate on how to call them. So that's why I said I was gonna use cheaters and exploiters interchangeably. But I do think that cheater has a connotation, like a moral connotation of like something that was supposed to do this thing, but cheated and ended up doing that other thing, right? I think I see that, that's how I see a cheater. And some people, including myself, try to avoid using the terminology and use exploiters, because it has, I think the word exploiter is more descriptive of what those individuals are actually doing. They are exploiting a resource or a service, not necessarily cheating. Like they don't necessarily were gonna be mutualistic and then decided to cheat. Sometimes they are never mutualists. They are always exploiters. It's like their species trait. Sometimes it's a behavior trait. But anyway, I think when we use the word cheater, I think there is a negative connotation, and I do think that it may be, I try to avoid using that word. But it's
why do you try to avoid that? Aren't there moral assumptions baked in, without, you can't really avoid it? Or why do you try to avoid it?
Because I think in nature it's also hard to talk about morals, because I think moral is a thing that's very from human nature. It's something that, and I think that would be in a philosophical discussion that I probably don't have knowledge enough to talk about that. But talking about animals or plants or bacteria or microbes that having moral, I think is putting an idea, a human being idea into those organisms.
But of course, the language we use is the story we tell each other. So so the whether you term them exploiters or cheaters, that's a story that's told to us about what is happening in the landscape.
Yeah.
Am I correct to assume that your research helps inform an idea of land management, or is that not necessarily an outcome of your research?
Not directly, but I think it has implications. Mm-hmm. I think because one of we are having a species loss with climate change, and that's something that we cannot really deny that is happening. Species are being extinct because of climate change and other anthropogenic disturbances. And that will have implications for communities, including those mutualistic communities. Because if, from my research, what we showed using those yeast model systems, we showed that with more species, that community is more resistant to the negative effects of exploitation. But if we are losing species because of other factors such as climate change, those communities are gonna be more exposed. That's not probably the word. More
vulnerable.
Yes, mm-hmm. to negative effects of exploitation and other factors that I didn't necessarily test. But
Jennifer, you had your hand up.
So I loved your talk, Myra, in part because I wonder what's going on with my microbe interactions in my own paintings. And in fact, I also have a very strong response to cheaters because I'm going to just tell a crazy story. I once lived in a co-op with a very, very, very funny socialist who never did any housework. And so you were doing the housework. And in my mind, he could have been a cheater because all he was doing was making jokes while I did the dishes and I swept the floor and I mopped the floor, right?
Mhm.
And so we could call him a cheater. But meanwhile, my ears were filled with humor. And so it was very rare that I would get really frustrated and be annoyed that he wasn't participating in the group project of our quote unquote cooperative. And so I guess what I'm wondering is, in sort of the reduction of the frame in which you are analyzing your systems, is it possible that in focusing in just a few players in this game, whether it's in your test tube or however else you're looking at it, that these cheaters are actually like little Robin Hoods that are stealing from rich systems and flitting off to another system and either dying there and feeding some other remote system, or leaving some unique molecule that only their body can make and no other body can make. So is this notion of cheating just a function of the scale of the system in which you're analyzing, as opposed to an understanding of the complexity of the resultant liveliness or vastness of potential genetic expression?
Yes. Yeah, this is a great question. I don't think I ever really thought of it that way, but I think you're right. And I think it's really on which perspective are we looking at. So if you look at the perspective of the interaction, of the mutualism, especially of that one player that is directly being affected by the cheater, then yeah, it makes sense to call this organism a cheater. But if you look at the, like you mentioned, like the cell will die, the yeast will die, and will release the nutrients that were inside the cell, and will probably feed the other organisms that are there. So I think it's, yeah, I think it really depends on the perspective, but and it can get pretty complex with only those, like I had like 8 species or 4 of each type, and I'm trying to think like how it affects it. The cheater affects differently one of those species and differently the other species from the same mutualist type. And it's really, yeah, it's really interesting. I think I'm gonna keep thinking about that, but I think you're right.
So the mutualism dynamic implies a relationship between two things, this party and this party. And so that relates also to a question that Nicole was putting out there earlier in terms of what is the total ecological dynamic here, and how do we consider that in this. But I want to get other people involved here. I want to turn to Mate and put this ethical question to you. And so obviously your presentation was the most explicit in terms of addressing ethics. And actually in response in a way to Myra, you're thinking about or thinking through a type of, so Myra said ethics is the domain of humans, or moral situations are the domain of humans, and you're proposing that maybe there's other forms of ethics. And I just want to actually you to clarify the ethical context in which you're working. So you brought up these ethical questions, but when you think about ethics, it's a set of principles that help guide an action. And I wonder if you could go back to your projects and describe what's at stake, or what is the ethical situation or the ethical dilemma at play in those projects.
Mm-hmm. Yeah, thank you. So I actually, so this was, in the presentation, it was sort of an abbreviation of like quite a multi-layered thing. And basically what I'm looking at is sort of connection of multiple approaches, multiple current trends or approaches, I would call it. So I'm looking at ethics connected with artificial intelligence. I'm looking at ethics connected with other living organisms than humans, and then I'm looking at ethics of humans and all of mutual interactions of these things or beings or whatever they could be. Sorry for lack of words. So, you know, if I'm and all of this is very intertwined, sort of in a feedback loop, because, for example, if you want to talk about and the newest research in machine ethics, which is, I would say, like the newest sort of newest discipline that is just emerging a couple of years now. So that is about ethics of artificially intelligent agents. However, they are thinking about it. People who write about it think about it through animal ethics, and so sort of the animal ethics came first, and then from there emerged is now like it's branching into animal ethics and something that is called machine ethics.
And you're talking about
yep.
an ethics among machines or among animals rather than ethics towards machines or towards animals.
So this is right now, it is at the moment is a little bit confusing, because machine ethics is actually on the crossroads of like exactly this. Like they're and I don't think they should maybe decide for one or the other, but I think that, you know, one approach is to look at how are machines acting towards other living or non-living agents, or the main question right now is basically, could a machine be a moral agent?
Mm-hmm.
And
what would the criteria for that be? Like, how would you know?
That is, yes. So you would know probably, like, so right now they're trying to figure it out. Like, this is the edge of that research, and there is no answer to it yet, but there are certain propositions. And, you know, as I was talking about the black boxes, so long before if you have a normal computer program, like old school style, where you can sort of read everything, go through it and see, oh, this is a decision it made at this point, and then there is branching into this decision and that decision, that's fine. But, and you know, but with machine learning, this is all lost. You sort of have a black box where you're looking only at the actions of that agent from the outside, sort of. It's almost like a behavioral observation more than anything else. So now the question is, what is it? You know, what is the difference between this type of behavior in artificial agent like that and an animal, for example?
Mm-hmm. Would you disagree with Myra that ethics and morals are the province of humans and humans alone, or do you take an agnostic stance to that? Do you not know yet, or wait, did you want to correct me? Am I misinterpreting?
Okay.
Yeah, go ahead. Yes,
no,
please.
I think maybe ethics and moral might be different things,
mm-hmm.
and I would go that moral is, in my view, and like we can discuss that further, but moral for me is this cultural thing. Ethics, I have a friend that's studying philosophy, and he studies ethics, and I will not go into that because I don't want to make anyone mad, but go ahead. Sorry.
So I mean, I'm sorry, what was the question again?
The question was, so I attributed to Myra the assertion that I guess in your language now, morals are the exclusive province of humans. Although my working definition of ethics is that ethics are a set of principles derived from morals that guide action. Maybe that's not exactly right, but yeah. But yeah.
So what I can say to that is that from the literature I have so far read, there is certain, like we can interchange moral agent with ethical agent. Like I don't really see the difference, and in the literature I have read so far, I have not observed a difference. People are using it interchangeably, kind of, but I mean, yeah, that's my thinking so far.
Too, is that it involves a decision about a right thing and a wrong thing, right? And so that would, so
yeah, that's why we say moral agent rather than something, well, rather than human or an animal or a bacteria or something like that. It's sort of, it puts them on the leveled plane, I would say, maybe. But yeah, I do not believe there is a really I mean, well, there is and there is not a difference. In actions there is no difference because everybody makes actions in the world. But you know, if you ask about who should take precedence, a machine or an animal, and if we reverse the ethics, like how that there should be some decision taken, then that one of them would suffer, who would you direct the suffering at, for example? You know there are these like ethical or moral dilemmas about, like, you have examples that I forgot the name of, like, it has a name, but it's basically you have a tram running on one track and you have an option, like you see that the tram is going to hit 5 people, and then you have an option to switch the tram to different track where it will kill only 2 people. Like, how do you decide? And so if we switch this example with animal and with machine, so that's, it becomes a little bit more crazy, a little bit more weird, a little bit weirder.
And does your work seek to reveal something about ethics and the differential between these potential ethics among animals and machines?
So I'm not sure what I'm going to discover. I'm moving forward, and we'll see. But I mean, so the current status is, for example, of this comparison, is that it is a sort of double-edged sword with the machine ethics, right? So like, are these machines up to certain point, like, responsible for some actions, which means that the human who stands behind those machines and is not responsible? What if this machine will create some sort of natural disaster? Who will be responsible? If this machine is owned by some huge company like BP, so now BP will be able to say, oh, it was not us, it was the machine who spilled it, so the machine should be held responsible. We don't want to pay whatever millions of billions of dollars for cleaning up. So this is like one of the things. But then the other thing is also, like, to what extent can we consider these things alive, which is also a valid question. And then it questions the definition of life, basically.
Yeah, which I want to get back to. That was a question that came up earlier, but I want to move a little bit. And also, I wanted to get in some of the comments in the side. So just on this current discussion, Caitlin, you made an observation. Do you want to talk about that, or do you want to just let the observation stand? I'd love to actually hear other people's voices if possible, if you want to. In terms of thinking about Myra's work vis-a-vis Matei's. Yeah, go ahead.
Oh yeah, I was just thinking of the way that we kind of the data that's used to build up the pieces to get to these larger models. And so thinking about Mayara's work as these kind of very intricate examinations of how mutualisms work, kind of, and how they respond when you perturb them, as the kind of basic data that you would need to build up this kind of larger ecological model with many interacting players that Jennifer was talking about, and how that really resonated with that work of using AI to build these black boxes of the interactions in Matej's work.
Great. Yeah, it's an interesting observation. Did Ty, did you want to respond to that at all, or should we move forward? It's an interesting observation about the relationship between the two works. I also wanted to get in, I always want to hear from Kathy. Are you there, Kathy? You asked, are there bacterial ethics? What was behind your question? And do you want to elaborate at all?
Yeah, I mean, I really love the 3 presentations. Thank you so much, everybody. They were fabulous. And, you know, I just was interested also in thinking about Jenny's presentation too, in terms of this kind of, I don't know if it's necessarily mutualism, but this kind of coexistence of bacteria that exists in these you know, industrial toxic sites. And so, you know, relating that back to I'm trying to relate that back to Myra's work, and then also to Matej's. So I'm sort of throwing out this very broad and kind of speculative question, probably, of whether there are bacterial ethics that we can think about, because I think that all of you are considering these kinds of, as Myra was talking about, these kind of mutualisms, which were so beautiful in the way that you put it, Myra. I really appreciate that. And just how we can consider these kinds of interactions in broader, maybe more philosophical terms, maybe scientific terms, but just terms that people can really wrap their heads around. Sorry, I'm getting texts somehow.
I was wondering, I was like, is that in the
Anyway,
chat?
blah, 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 blah. You get my message. Anyway,
Yeah.
I hope that's clear. Thank you all.
Does anyone want to jump? Jennifer, you unmuted. You want to jump in on that?
Yeah, at first I also want to say I realized I didn't acknowledge RPI, which supported work last fall. So like, Kathy,
Kathy's
just awesome.
listening.
So, yeah. So I think the reason why I am so compelled by the bacteria is that I am trying very hard to understand ethics through the bacteria, meaning I'm trying to think about how on a finite earth you can have infinite expression. And what gives me so much hope in those paintings is that somebody asked a question in the thread earlier, like, do these change? My gosh, these things never stop changing. And the glory is the color. But then if you see a new color, that means an old color got destroyed. And so what is the ethics of color transition in my paintings? And I am sort of falling into this notion that I don't believe in justice and I don't believe in ethics. What I believe in is a diversity of expression, that life is actually, or maybe not even life, like material, like the different formulations of chemical materials. The glory is in the diversity of those compositions of the elements in the periodic table and the various ways in which they interact. And the more that we can spawn that diversity of expression, the more we can understand the potentiality of the earth. Because I really don't think that, you know, my ethical attempt at eating a fair trade orange, I'm not convinced that my life is at all ethical. I have to destroy necessarily in order for me to proceed. And that destruction happens all the time in my paintings. And yet when we step back and we look at the performance of all of this creative expression, we see a kind of glory that is predicated on destruction. And
Just to, that is a very beautiful thought.
So,
I don't mean to pass that.
again, I'm not.
Oh, I'm sorry. I didn't mean to cut you off either. Are you? That's a very beautiful thought, and I wanted to linger on that, but linger on it by pushing back against it a little bit, just in the sense that you began your presentation with a discussion of climate change mitigation efforts, and then moved on to the paintings, and then talked about how these might synthesize these two different, very apparently different methodological aspects of your professional life might end up merging. But it occurred to me as you're talking about that, even the very notion that we should be addressing climate change has to stem from some sort of, let's call it, position of care or concern. And it's hard for me to separate that from an ethical question. And one of the interesting things about a position of care is that it's a kind of time folded. That is, you're thinking of, you're trying to repair something, so you're thinking about the future, already thinking about the future, so already making calculations about the future and thinking about a desired future. So it's hard for me to see that as not an ethical maneuver. And I was struck actually in this connection, and how what the question on ethics I wanted to ask you is actually sort of a personal question, which I hope is okay. I was fooling around the internet looking up each of you before this talk, and I heard a presentation of yours, Jennifer, where you said that you went through a period of despair about climate change. If anyone on this call hasn't gone through that period of despair yet, I recommend it. It's sort of like essential, I think, and I've been there myself, and I kind of wondered how you came out of that and how that's affected your work.
Well, I frequently return. Like right now, I am furious about New York State's initiative. I cannot believe I'm furious about it because I've been so pro greenhouse gas mitigation and solar panels, but one of the outfalls from the policy of solar panel and renewable energy in the state, which again, I totally support, but what's happening is it requires a certain level of the electric grid to be established. And so as a result, some prime agricultural land is now housing solar panels or is scheduled to house solar panels. I love agriculture. I love solar. I love greenhouse gas mitigation. But the fact that we have a grid that goes right next to prime agricultural land means that we're going to lose that prime agricultural land to solar. There's a lot of thought, there's a lot of care in this attempt to address the climate change, but it's almost like I wish we would have a bit more of a pause to make an integrated decision. But again, we've had the last 20 years to make an integrated decision. And now we're coming up to this 10-year leap before 2030, which I'm really proud that we're trying to take. But it's as if we're constantly behind the curve. So I don't know, I guess in my mud paintings, to sort of get back to the poetry of it, I mean, I'm giving you that sort of concrete example that's happening right now, because there isn't a crop a farmer can make for $1,200 an acre the way that you can make $1,200 an acre from solar. But that land is very precious, and we should think more smartly as a collective community if we really care about the whole system. So I'm contending that care is positional, like it isn't universal. But in my mud paintings, what makes me so happy is that they are endlessly self-recreating. And so what makes this sort of a living dynamic system is actually the giving up of a certain amount of agency to give those resources to something else that then becomes the next generation, which is kind of what's curious about Mate when you were talking about how understanding is the ability to, did you say recreate? What did you say? Understanding
Build.
is the ability to build. And what I think is happening in my mud paintings is understanding is the ability to unbuild or decompose, because in the process of decomposition, you have all of those resources available to make the next generation of thinking. And so if I were to go back to climate change with that context, I would say that, okay, we're going to build solar panels for the next 25 years on that land. And then maybe after those 25 years, we'll do something different on that land. And that's sort of how I get my optimism back, because every decision that we're making is setting the stage for the next set of decisions. And really, it doesn't really matter because things live on planet Earth. Whether or not it's human, it's another matter. I don't know. I don't think I was too optimistic there.
No, I like that. It's that's another way to think about it. The construction or unbuilding, and yeah, it's whatever we do. The bottom line is whatever we do, it has consequences always, no matter what it is. So there is, you cannot do not consequential thing probably. Yeah. So. But the comparing, you know, the understanding with ability to build, it does not has necessarily to happen, because like for example, I meant it only in a particular way. How I think it was Loeb who said that. That's the engineering paradigm in science. You know, that's the, I mean, and I'm sorry if I'm completely wrong, scientists correct me, but you know, not all science has to be able to build. You can make observation and from there you do your research instead of building stuff. But he, Loeb, for example, said that like he does not, everything else is metaphysics if you cannot build something out of it. So should be dismissed. And that basically means that, you know, I mean, a lot of stuff would be dismissed we have now.
A function of a definition of understanding that is action contingent is a strange idea to me. You know, Plato, in his account of Socrates' wisdom, for Socrates, pure thinking, pure contemplation, was thinking without even writing, was thinking without any action whatsoever, and that Plato felt himself in a bit of a compromised position just because he was writing down, so he was less than Socrates just by that notion. And I think there just has to be a form of understanding that would encompass that actionless comprehension. There's a thinker I'm very interested in named Edouard Glissant, who distinguishes between two forms of understanding in French. He writes in French, and one is comprendre and one is donner avec, which is a neologism he invented. And comprendre is a very acquisitive form of understanding. It's like taking. Prendre is to take in Latin, and take, you know, to take and grasp and hold, as opposed to his idea of understanding, kind of poetic understanding or poetic knowledge, which is donner avec, which means to give with and towards, sort of a yielding type of understanding. I would hope that we're, in my ethical formulation, I would like to be thinking we're building a society that can encompass both forms of understanding. And I feel like every one of the presentations actually does have that going on in them.
Because yeah, once you build something, you change the understanding, and it, you know, you influence, same as when you're building experiments that they are constructed, which, you know, excludes some kinds of knowledge and invites some particular other, I guess. So, and I mean, scientists have training in that, like they know a lot, but still, if we keep that in mind, it's yeah, there's a lot of construction going on, constructing apparatuses, constructing methods, constructing, you know, constructing research. Everything is actually made. I mean, so how, and that those actions, we like the building, we do sort of translates what we see. or influences what we see.
Did you say everything is? Oh, go ahead, Jennifer. Yeah.
I just want to say that last fall I took a course on geobiology, and it blew my mind because there's billions and billions and billions of years where there's no life on planet Earth, and yet there's all these different forms. And that's why I not that I'm bored with our human stories. I'm not bored with our human stories. There's so many of them, and they're all interesting and crazy and fascinating. But what is just amazing is, like, in people who are thinking about the origin of life, one of the things that they're looking for is a abiotic process that self-replicates. So like a molecule that causes another molecule to be formed in order for you to have a concentration of said molecule. And that makes a microecosystem, in which case another molecule can self-form and self-replicate. And this idea of replication as being this opportunity to acquire a kind of knowledge or knowingness of an ecosystem, like without human morality, without this notion of scarcity. And I guess I'm just throwing that out there because I actually, at a FEM meeting where I met Kathy, there was Kathy, if you're still on, the name of the woman, the philosopher who was talking about sort of the deservingness of abiotic life forms. And there's a whole solar system out there, and I don't care if there's not life on that solar system, because if you look at any one of their local planets, the ecosystem on that planet is so different and diverse. And so I'm kind of wondering if we are, I guess by nature of our desire to survive, sort of fetishize this sort of ethics of beingness, when in fact an earthquake could gobble us all up, right? So
It's anthropomorphized. Kind of.
Fetishized or anthropomorphized. I mean, those are almost equivalent, right? In this case, but I do wonder how that train of thought squares with your very felt and intense drive to mitigate climate change. Why not just let the system unfold as it will? There's entropy in the system; it'll take care of itself. And
because I like acts of futility.
That's a cop out. But
No. Okay, so here's the real reason. It took so long to have this diversity that we have, and it happened because we had this pretty stable climate. And so it's not about whether or not McMansions on the ocean get flooded and the rest of us have to pay for the insurance of that. Like, I'm being a little cynical here. It actually has to do with the diversity of the animal species and the plant species and all of that. I have full belief that the Earth will have resilient life that continues. It's just like in every previous disaster, we lose a diversity. And what's so great about a baseline of diversity is that you have all these new books that come off, like all of these successors that come from the genealogy of those that came before. And if we kill them all out because we change the climate so much and they can't adapt fast enough, well, then we lose that expressiveness. And I think that's fundamentally where my ethics are, is sort of honoring that expressiveness that is so unique to our planet.
And I think that expression of, I'm just going to call it love. You can be embarrassed by that if you want, but where that comes from, and Matei, I was thinking about that during your presentation in terms of, you can model the structure of life or the structure of growth or the essence of growth, and you can talk about that accumulating and so on and building. But at some point, there seems like there's a necessity for some sort of inspiration in it, and I don't know how to account for that or name it.
Yeah, I rather criticize people who would think that they can model the essence of growth or the essence of life. I don't think I don't think this is correct. What they are like it's. I mean, or there are certain parts that are right, but it's not all. And that's interesting that we actually create and build organisms and things out of that, even though they are incomplete. They're by definition they're incomplete because the models are incomplete we have. Yet I'm kind of trying to raise awareness in a way about this discrepancy between the model, incomplete model, and considering it actually life. I mean, you know, with open mind, it could be maybe somehow different, but also it's definitely not the same as the life we have. And that yeah. Sorry.
I'm sorry. No, I'm sorry. I came across an observation in an essay this morning. I don't know who to attribute it to. I can't remember, but it was that consider that human beings could be a pollinator species for like a machine life to come.
Yeah, that's the transhumanism / H+. And it's like Elon Musk and etc. Yeah, it's Ray Kurzweil. It's there is this idea that you know, like machines will supersede our human intelligence, and not only one time, but like many thousand times, and then you know they can destroy the life on Earth. And I mean, I can see that option. But I don't think it's about intelligence, or I don't think, you know, there is again the word intelligence, but is intelligence the measuring stick? Should it be for life? I mean, you know, what is more successful living species? Are those humans or bacteria? There is much more bacteria than humans around. Plants, again, the same stuff. Like, I mean, it's just the way how you look at it, from what angle.
Bacteria might be like happier. They might not be so obsessed with lack.
Who knows?
Jennifer, you had your hand up. Sorry to intercede.
No, I'm gonna stop or not say anything. It was something Matei had said earlier, and then he sort of went there. The idea of life that is intelligent would not kill other life. I mean, obliterate other life. So this fear that we develop a more intelligent life form than ourselves. I mean, and I'm not suggesting that we are the most intelligent life form by saying this, but the fear that if we created a more intelligent life form, that life form would destroy us. I think by definition, that would not be a more intelligent life form, because a more intelligent life form would have a lot more respect for other kinds of life forms. And so, I mean, that's my personal judgment call, because you can go kill things. It's much harder to coexist. I mean, you can get rid of.
I mean, I was thinking about that when I was listening to Myra's presentation. I was like, these exploiters seem pretty stupid. Like, they're destroying their own environment and ecosystem. It's like a kind of a dead end job, I think, is what I wrote in the margin.
It's again the words. It's like you know, intelligence or exploiters. I don't think it is the same meaning of intelligence these people are applying to machines than to humans, and like all of for them it's only ability to react or adapt. It's not really anything else. So I mean, then again, like, why are they calling it intelligence even? Should it be called something else? Or same as with the bacteria. Like, I mean, there are no single meanings for particular words, unfortunately, and people are exploiting those meanings, different meanings.
I think some of this, there was a comment earlier, some of this is like the foundations of science fiction, right? Like, it's like, I don't know, part that you were talking about before, Jennifer, like why would, if we create a more intelligent being, would they kill us? And it's like the basis of Battlestar Galactica, where they created artificial intelligence and these like robots, right, that mimic us, and then they killed us all, and then whatever. Like it's the whole basis of the storyline. So I don't know if that's just what we're known, like that's what we grow up thinking.
Or as a reflection of how we do think. I think,
Yeah.
I mean, because I'm just, it's a comment on intelligence and how we define intelligence, because there's plenty of historical precedents of plunder, you know, brawn, and I'm not saying that there isn't a place for brawn in this world, but
Yeah.
I don't know. I didn't mean to, I should have kept my hand down.
I liked it. I thought it was a good one.
It's I mean, just very quick last thing maybe about it is that I see this more like from this technology community, it comes out more as sort of similar to the fear of gray goo, which was the fear about nanotechnologies, that it will keep replicating, just like it, not even intelligently, it will just like sort of keep doing its thing, and that will kill us. This and not regarding anything around it or anybody around it. Yeah,
Is that a metaphor for us?
Yeah.
I mean, I'm just saying, like, we talk about invasive plant species, but we're like the biggest invasive species there is, right? Like, we talk about, you know, deer destroying our forests in upstate New York, but like, I don't know. I'm gonna stop. I'm getting very whatever.
Well, we probably at some point should wrap the evening up.
I was just gonna make that maneuver. Yeah. So
Looking at you.
not that I could talk to you guys all day, but it's Zoom. We're all on Zoom.
Awesome.
Zoom, more and more Zoom. This is very delightful. I should, before we close it up, though, does anyone here have anything that was on their mind that they had been?
I have been trying to formulate my thoughts, but I'm just extremely tired, and I'm almost sure that I'm not going to be very clear. But because there was this conversation earlier, what is the difference between moral and ethics? And I think throughout the conversation about justice and the conversation about they are going to destroy us, so this kind of a dualism, this is where, or what Jennifer was talking about justice, and then the attitude towards climate change and how decisions are made on governmental level for what reasons. This is where we see where all these discussions, they come together. We see the difference between ethics and morale. To me, the way how I understand it and the way how I have seen this used in sociology and anthropology or even philosophy to some extent is that the ethics turn into moral when we make a law, and this law becomes something that we should accept as our identity, collective identity, national identity, group identity, whatever. And when this becomes a necessity, this is already the cultural side of it, a necessity of if you want to belong, then you have to follow this ethics. Then you have to think about the other species as invasive to you. And if you want to belong to a society or community that thinks that way, then you obtain this moral, then you keep it and you proclaim it, and you force the other people who are part of the community to think about it as their own identity. And ethics kind of flows. This is the discussion about how we deal with so many different ways of interaction, which for me, it flows from one community to another community, or it crosses borders. So in a way, moral is this way of cultural event, which is obligation. It becomes an obligation, an ethics that became an obligation. So I just wanted to mention this. And so to some extent, when we were talking about the science fiction, it's like we have the obligation to think about the robots as somebody who's gonna kill us. we have to think that way. We should not think about the robots as somebody who can be our friend, or maybe we just like them to kill us. This is what maybe we just like to be killed. We want to be destroyed for some reason. But no, we should think about the robots as somebody who may outsmart us and kill us at the end, and this is threatening, so we need to be very careful.
And
so
that is,
yeah,
it's so pervasive how much that anxiety plays out in cinema. That, you know, it's almost every Star Wars, every film is like the machine is going to come kill us.
Yeah, I really.
Whoa! Who said something about Star Wars?
There was good robots in Star Wars.
True. C-3PO was very pleasant chap.
Yeah.
But
yeah,
you know, like as soon as you become on the dark side, you become mechanical in some way. Luke takes a step to the dark side, gets a mechanical arm. Darth Vader becomes evil; he becomes entirely mechanical. I'm just saying.
Yeah.
Yeah, that's the fear of the uncanniness of the machine. But I just wanted to mention something which I'm sorry that I missed Matej's presentation, but I'll watch it later. But because I've worked a lot on, I did performances and stuff related to artificial intelligence and ethics and stuff. And so the first time when we talk about an intelligent machine or a robot is the Czech writer Karel Čapek and his brother Josef Čapek, who suggested the word robot, which comes from the Slavic word work or rabuta. I don't know how, yeah. So, it is about machines that are created by us to do the job that is unpleasant. And then suddenly they became independent and they kill us. So this is like from the very first moment when we started to think about the cultural implications of our the machines that we create. This is what we kind of started thinking about. They're gonna kill us.
I would just add to that this play, Rosen's Universal Robots, was actually the first time these were not mechanical robots. Robots in the original versions were made in a test tube out of chemicals and organic material. So it was the first synthetic biology.
I forgot about this detail. Yeah. Mm-hmm.
But people at that time were not able to really like take it as their own, so they changed it into something mechanical because that was easier to understand at that time. 1900s, sort of.
1920, I think.
1920, okay.
1920, 1926, something like that.
Early, yeah.
Covered some ground. Nicole, you had unmuted at one point. Did you want to say anything, or did the moment pass?
Yeah, so I was just really interested by all three talks and how it seemed that no matter who the speaker was, there was this sense of interactions. So Myra talked about mutualism, Matej talked about interactions with or without machine learning, and Jennifer talked about interactions within microbes. And I was wondering if any of the speakers kind of looked back after they gave their presentation and looked at the interactions between the other speakers and if they learned something about their own project itself. I know it's super broad, but pretty much where I'm starting.
That's a great concluding question. Thank you. Yeah.
I will just quickly say that this conversation about work, I think very much connects with Myra's mutualism and my mud. And also Matej and your sense of understanding. Because I do think that the process of makingness is the true genesis. Like the doing the work is the true genesis. And so if the robot does the work, well then go. right? Like if the microbe does the work, like in Gowanus Canal, people are, buying property there, speculating that the property will get clean. Right now, I consider it a true wilderness because humans don't want to go down into the Gowanus. And so it's sort of a pristine landscape for organisms to take part, and they do the work of living in the Gowanus Canal. And so I just think it's really interesting how this conversation started out about cheaters stealing resources from, and not contributing, and then ended with robots who actually do all this work and then are like hostile because they're getting no benefit from doing their work because there are these cheaters called humans that use them to do work. And so I guess what that makes me realize, if I reflect back on my own work, that this idea that I'm obsessed with of diversity of expression is fundamentally born out of work. And so those that work are those that activate the finite materials of the earth. Whether or not that's ethical, I don't know, but I like thinking about how my microbes are making these proteins. They spit them out into the external spaces. These enzymes in the external spaces chop up all of the natural resources, and then any microbe in the proximity has the capacity to soak up those broken down resources in order to incorporate them themselves. And that's a kind of collaborative work. I don't know, I'm rambling, but it's late, and that's kind of how I'm thinking about Myra's and my talk. And Matei just disturbs me. Sorry, Matei, I love you, but like,
I'm sorry I had to switch my phone because battery ran out.
So you missed what I said. That's good.
The last sentence, I missed your last sentence.
She said, Matei just disturbs me.
Oh, no.
I believe I got that right. Is that a quote?
I can't remember, I'm sure.
She said, Matei, I love you, but
No, I really like for me, the biggest takeaway will be now I'll be thinking about ethics of microbes. I mean, for real, it's interesting. It's super interesting. For some reason, I have not came across that too much, and it's worthwhile to think about it.
Yeah, for me, I think I'm still thinking about Jennifer's question about cheaters actually being the Robin Hoods. So I'm gonna be thinking about that, and for sure. And I also, yeah, I think I need to, yeah, I'm gonna be thinking about the ethics of animals and ethics or moral of animals, or non-human things, non-human life, I guess. This was really interesting.
Yours as a final parting thought. That's a really nice way to end. Heidi, did you have any? I thought you had raised your hand. Did you want to wrap up our proceedings formally?
Yes, I think it's time to wrap up. Thank you, everyone. And I loved all the talks. I really loved the discussion this time. It was great. And yeah, hopefully see you all at the next BioArt.
Yeah.
Thank you,
Thank you.
everyone.
Thank you.
Thank you, everyone. I'm sorry I wasn't there in the beginning, but yeah, thank you. It was great.
Thank you. Bye-bye.